the main things that I hope you take away from biochemistry is the meaning of and the importance of the KD, or the binding dissociation constant. It's a measure of binding strength, or what we call affinity, but it's actually the inverse of the binding affinity, Ka. Not to be confused with the acid dissociation constant, Ka. I know, we make things complicated. But basically, the reason why we use Kd instead of Ka, that is the association constant, is that the association constant has units of like 1 over molarity, or 1 over the concentration, which that would actually not be very helpful. So the reason why we use Kd is actually to be helpful, because if you use the Kd, well, now what you're talking about is units of molarity or units of concentration. What it boils down to is that the Kd is going to equal the concentration at which half of one partner at which half of its other partner is bound. The lower the Kd, the higher the affinity, whereas the higher the Kd, the lower the affinity. Basically, if you need more of the other thing around in order for half of you to be bound, this means you have a lower affinity and you're going to have a higher Kd. It's going to take more of that other partner to find half of you bound. Whereas if you have a lower, a stronger binding affinity, then there needs to be less of the other thing around for half of you to be bound, and therefore you're going to have a lower Kd. So a smaller concentration of that other thing is needed. We'll get into all of the math, but first let's think about it a little intuitively. Let's talk about taking a molecular census. So we're going to take these two binding partners, we'll call them A and B, mix them together, let them get married, that is bind, and divorce, that is unbind. And then we'll go and we'll see how much of them are married and how much of them are single. Now I'm going to use the terms A and B, but you might see different terms used. Your book uses the terms P and L. So basically, L stands for ligand. And so a ligand is just a generic name for a binding partner. We have more specific terms that we can use for ligands of different molecules. When we talk about antibodies, say, we'll talk about antigens. When we talk about enzymes, we'll talk about substrates, which are the ligands that enzymes bind and modify. But ligand is more this generic umbrella term. You'll often see it used in concert with R, or with a receptor. So sometimes you'll see R for receptors. Basically, these are going to often be membrane-bound proteins, but not always, and they're going to bind to signaling molecules, and those signaling molecules would be their ligands, and then they're able to transmit the signal. So you'll sometimes see R used, you'll sometimes see R and L, you'll sometimes see P and L, so your book uses P and L, so for protein and ligand, but it doesn't have to be a protein. It can be other things too, and so we can write this same sort of equilibrium constant for whatever our A and B are, and so I'm just going to use the terms A and B. Now, A and B are going to have some intrinsic affinity for one another. And of course, as we'll see, it's going to depend on like their surroundings, like salt concentration, pH, all that good stuff, because nothing's ever simple in biochemistry, right? But they have some inherent probabilities of binding and unbinding. And so we can talk about binding when we talk about them binding, or like getting married, as I like to think about it. Here, what we're talking about is the forward rate of this reaction. A plus B goes to AB. So this is going to be our K on. Your book refers to this as Ka, but in little ka. So we talk about a rate constant. So like one of these rate constants, we're talking about like a lowercase k. And if we're talking about an equilibrium constant like a Kd, our binding dissociation constant, or our Ka, our binding association constant, that's going to be a big K. So your book uses little Ka and little Kd, but I think that's really confusing. So I'm going to use K on and K off. Now, K on is going to kind of like present how likely they are to actually get married if they bump into one another. So if they bump into another, how likely are they to get married? So you can think of this as kind of like that whole like attraction at first sight. So maybe you see them and you're really, really attracted to them. That would be, you have a high K on. But so maybe you get married and maybe it really is love at first sight. And so you'd have a low K off. You'd be less likely to get divorced, not very likely to get divorced. You'd have a low K off. But say he's not really what you thought. And so maybe now you have a high ability of
K of divorcing, and so that would have a high K off. That would then counterbalance the K on, so that when these molecules reach equilibrium, that is when the same number for each couple that marries, there's a couple that divorces. Basically, what's going to then happen is you reach this dynamic equilibrium, and the K base that's going to be based on the K on and the K off. So we can actually write the KD, which we'll talk about more in terms of concentration, but we can also talk about the KD is equal to the K off divided by the K on. So bottom line, these rate constants, these little K things are going to influence how much of them we find at how much of each of these we find at equilibrium. And so because of this, we're able to then use the concentrations that we find at equilibrium in order to tell us about the affinity. But it's not going to be able to tell us about the relative contributions of K on and K off. It'll just tell us about kind of like how it all plays out at equilibrium with some balance of the K on and the K off. All being equal, if you were to increase the K on, you're going to decrease the KD. And if you were to decrease the K off, you would decrease the KD as well. And so hopefully this will make more sense when we talk about KD in terms of concentrations at equilibrium. Just remember that those concentrations at equilibrium are coming from how likely they are to get married if they bump into one another, and how likely they are to get divorced if they get married. What happens is you reach an equilibrium where for each one that gets married, there's a couple that gets divorced. And so the absolute numbers on either side of the equation aren't changing. So let's think about a couple scenarios. What if you had higher concentrations? Well, at higher concentrations, you're more likely to meet. So there are more opportunities to get married. But you still have the same probability of marrying each time you meet. So basically, the amount that you're attracted to them at that first sight, that's not going to be changing depending on how many of them you see. You're just going to have more opportunities to see them. But that also means that you're going to have end up having more married couples, which means there's more opportunities for divorce, because you remember, you kind of have that probability of divorcing, that intrinsic kind of probability of divorcing. And so this means that at the higher concentration, you get more marriages, but then you also get more divorces. And so that's why you reach this kind of equilibrium. And then what we can do is, once I've reached that equilibrium, we can take our molecular census. We can go and we can look at how much is bound and how much of it is unbound. And we're going to do this by using this KD, this dissociation constant. So this KD is going to be the dissociation constant. And we'll look at 1 second at where it comes from, but hopefully it's reminding you a little bit about when we were talking about our equilibrium reactions in terms of acids and bases, and how we could use the law of mass action, or I guess maybe I didn't call it that, but basically we can write this equilibrium constant for any sort of reaction. And this is going to be the equilibrium constant in the reverse direction of how we have it written above. And again, I'll show you this in a minute. But for now, just know that we take this molecular census, and what it boils down to is that the KD is going to tell us how much B we had to start with for half of the A's to be married when we go and we take that census. So let's think about what would happen if we really if we had a high affinity. Well, if we had a high affinity, we don't need to see as many of them in order to actually have half of us be bound, because we kind of we really want to marry them if we see them, and so we don't need to see as many of them, or we're less likely to divorce if we marry them. So even if we don't see that many of them, each time we're married, we are going to stay married for forever, love forever, not just love temporarily. And so that would be an example of how you would get a lower KD. But say you don't like the other, say you have a lower affinity, maybe you aren't as likely to get married, maybe you're more likely to get divorced. Either way, you're going to have more of the other partner around in order for half of it to end up being bound. And so what this means is basically we have this way to connect the concentrations of a binding partner to the to the strength of the interaction. And this is through the KD. If we have a lower KD, this is going to be a higher affinity. And so in this example, the blue line would have a lower KD, it would have a higher affinity, whereas the orange line would have a higher KD, a lower affinity. And note that basically, when we're looking at a graph like this, this isn't telling us anything directly about the K on or the K off. It's just telling you about kind of like the end result of the two. But it's telling you this useful value that we can actually kind of think about intuitively, and we can actually measure in the lab. So let's talk a little bit more about where these numbers are actually coming from. So remember, we had that generic form where we had A plus B, or we had little a, B plus C plus D. And then we said, okay, well, we could write this equilibrium constant where we had C to the C times D to the D over our A to the A and B to the B. So basically we had our products over our reactants. And we could do this for any equation that reaches equilibrium. Now let's think about it in terms of this reaction, of a binding. So now what we have is we have A plus B is going to AB. If we write our equilibrium constant for this forward direction for the binding, what we get is the Ka. So this is going to be the equilibrium constant for binding. Okay, so why don't we use this directly? Let's think about this. Say each of these is in constant, say we have units of molarity. If we have this Ka, so what we think about is, okay, that's in molarity, this is in molarity, and this is in molarity. And so what this is going to happen is those would cancel out, and now you're left with units of 1 over molarity, which isn't very convenient. And note that I'm using molarity here, but it could also be, you could also be using like millimolar or nanomolar or something like that, some sort of unit of concentration. And you end up with 1 over that unit of concentration. Now, well, it's 1 over that. So if we just take the inverse, we'll get units of molarity. So that's what we'll do. We can take the KD as 1 over the Ka, and this will give us units of molarity or units of concentration. What we're doing, we saw it here just by taking the inverse of the Ka, but conversely, we can also write it by just rewriting our equilibrium equation. So instead of thinking of it from the perspective of binding, we think of it from the perspective of unbinding. Well, now what we're saying is that we have something that looks more like this. And so now we can go and we can write that equilibrium constant just like we did before, but here we're going to have our binding partners, the unbound versions on the top and the bound version on the bottom. And so this is how we get to the KD, and this is going to be the equilibrium constant for unbinding. And again, it's going to have units of concentration. And so that's going to make it really, really helpful. And remember that here we're going to have a big K. And here we're going to have, with our rates, we're going to have small k's, and they're typically going to be italicized. So that's our KD. And now let's think about what does and doesn't influence it. So let's think about the concentration, changes in the concentration. So what if people move in or out of the area? Well, as we talked about before, this means that people are more or less likely to meet potential partners. So you're more likely to find a potential partner if people move in, and you're less likely to find a potential partner if people move out. But you don't like those people any more or less than you did before, and so KD isn't going to depend on the concentration. You're not changing those rate constants, so you'll reach the same ratio at equilibrium, and you don't change the KD based on the concentration. But you are going to change the amount that you find bound of those partners, just not the ratio of them. And this is going to be a useful relationship that we can use in order to actually calculate KDs from binding measurements. So the concentration will not affect KD, because remember, that's going to be this ratio of your A times your B over your AB, but will affect amount bound. Because that's going to actually depend on the concentrations, how likely you are to be able to find one of them. And so if there's none of them around, you're not very likely to find them. You're not going to find these molecules bound to one another. And if there's a lot around, well, now you're more likely that you'll find them bound. And even if they are to have a high divorce rate, a high payoff, there's still going to be lots of them around that they can bind yet again. So the amount that's bound is going to depend on the concentration, but not the actual KD, not that intrinsic ratio that they'll reach once they reach equilibrium. And so this useful relationship is going to allow us to calculate KD by taking measuring binding, measuring proportions bound at different concentrations. So KD isn't going to depend on the concentrations, but it does depend on that K on and that K off, as we talked about. So remember that our K on, if the partner's more attractive, each time they meet, their chance of marrying is higher. And so there's going to be more married couples when you go take that census. Your KD is going to be lower. Conversely, if they fight a lot, if you have a high off rate, a high divorce rate, then there's going to be fewer married couples, and so that is going to matter. So your KD is going to depend on your K on and your K off. But again, if we just measure the concentrations, if we just measure the concentrations that are bound, that's not going to tell us directly about the K on versus the K off. There are experimental methods that you can use to actually determine those, but when we talk about thermodynamic measurements at equilibrium, there we've already lost all that information. In terms of some actual values, so KDs of some typical types of intermolecular interactions that we're talking about in biochemistry, they kind of span a wide range. So the strongest non-covalent interaction that we know about really is this biotin-avidin, which has this 10 to the minus 15th affinity. Crazy, crazy low. When we're talking about other types, more common affinities, we're talking about things like nanomolar, micromolar, millimolar would be low affinity. So remember, 10 to the minus third, this is going to be our millimolar. If we're talking about 10 to the minus 6, this is going to be our micromolar. 10 to the minus 8, that's going to be our nanomolar. 10 to the minus 12, that's going to be our picomolar. And now we're talking about all the way down into the femtomolar range, which is really, really, really ridiculously tight. And because remember, the smaller the value, the tighter the binding. And so you can see enzymes have micromolar range affinities typically. Antibody antigen, we're talking about picomolar, really strong ones to nanomolars. Typical receptor ligands, we're dealing with nanomolar to micromolar. So these are some of the common kind of ranges that you're going to see. So we said that we have that relationship where KD equals concentration of one binding partner at which half the other is bound. And we'll look mathematically at why this is in a minute. But basically, we have this connection between the concentration of one binding partner and the probability that we're going to find the other partner married to it. So if we can measure the proportion of B bound, which we often abbreviate as theta B, over a range of A concentrations of A, we can then calculate the KD. So often what this means is practice is you would say make a serial dilution of A. So when we talk about serial dilution, we're diluting something by the same amount, by the same dilution factor over and over and over. So we can span a wide range of concentrations. And then we have a constant low amount of something that the measurable B. And so we'll get more into a minute about why this needs to be a low amount, but it does. And so we have a constant amount of one partner, a variable amount of the other partner, and then we mix them, we let them reach equilibrium, and then we take that census, we see how much is bound and how much of it is unbound. Now let's think about what would happen at different points along this measurement. On our x-axis, we're going to have our concentration of A, and on the y-axis, we're going to have the proportion of B that's bound. And so remember that this is sometimes theta B. Okay, so now if we have a little, not very much A, say if we have no A around, then none of B is bound. And what's going to happen as we start having more A? Well, now some of your B is going to be bound. Initially, you're not going to have that much bound because there's just not enough A. But now what's going to happen is you're going to get more and more and more of it bound. But what happens if you end up with a lot of A? Well, now all your B is kind of going to be saturated. So if you have a ton of A, you're going to saturate all of your B. And so what's going to happen is that you're going to be all bound. And so even if you add more A, you're kind of going to plateau out. And so this place that we plateau out, and then in between, we'll kind of have a curve like this. So we kind of get this hyperbolic curve. We can give different names to these points. So basically this amount at which we have it all bound, sometimes we'll see this as like B max. And then we have this 0, so that's none bound. And then remember our KD, that was the point at which half of it is bound. So if we say, okay, 1/2 B max, this is going to be, that would be the proportion that's bound. And then we look to see the A concentration, and that would be our KD. So we end up with, draw more nicely, something that looks like this. But you'll actually often see them drawn something more like this. So where we have a semi-log plot. So basically it's a semi-log because your x-axis is in log, so this might be log 10 commonly. And then your y-axis is going to be linear scale. And basically the advantage of this is you're kind of spreading out points that get clustered in the beginning. So here is some actual data from grad school I took. And I did a ton of binding measurements, and you can see that I have all these points that are kind of clustered down here in the low concentrations. So we can't really see very well. But if I graph them on a log 10 scale, well, now these are spread out. I'm able to see the different points more on my scale and see that something weird was happening when I was measuring. Now, you always want to make sure that you're looking at the axis. So look at your x-axis. See if you are in log or if you're in linear. If you're in linear, you should see a hyperbolic graph if there's a single site. And if you're in log, you should see a sigmoidal. You might see a sigmoidal if you're in linear scale, if you have something that has multiple binding sites and shows cooperativity. We'll get much more into this when we talk about hemoglobin. So hemoglobin, it has 4 binding sites for oxygen, and it binds cooperatively, which means that when oxygen binds one site, it makes the other sites be more likely to bind. And so even though it's linear, you're going to get the sigmoidal curve. If you were to go and you were to plot it on a log scale, you would get an even steeper sigmoidal curve. But just know that a sigmoidal curve will only indicate cooperativity if you are in a linear scale. And you can't tell if you're always going to see a sigmoidal curve if you're in a log scale, even if there's a single binding site. And so again, we'll talk about this a lot when we talk about hemoglobin binding, but just be aware that you always need to look at your x-axis. So just to review from this first part, we have the KD is going to be the dissociation constant, and it's equal to the concentration of A times the concentration of B over the concentration of A times B. So you have your individual molecules, the unbound times the unbound over the bound. Remember that this is going to be the inverse of the association constant, that is the bound over the unbound. And so what this means is that the smaller the value of the KD, the stronger the affinity. The higher the value, the lower the affinity. So if you have a high affinity, what this means is that there doesn't need to be as much of the other thing around in order for half of you to be bound. This could be because you're more likely to get married if you find them, or it could be that you're less likely to get divorced when you find them. If all we're doing is looking at the equilibrium amounts, we can't tell anything about those on and off rates directly, but we can see like kind of like the ratio of the two, because remember our KD is also going to equal our K off over K on. But when we do equilibrium thermodynamics, we're not going to be able to tell contributions of K off versus K on. There are methods we can use, but not the equilibrium ones. What's going to happen if you have a stronger affinity, a lower KD, you need less of that other partner around. But if you have a weaker affinity, well, now you're going to need more chances to run into them. And so you're going to need a higher concentration of B in order for half of it to be bound when you were to go and look at equilibrium. Remember that these values typically range from often they're ranging in like the nanomolar to micromolar scale. I might be a little biased in the importance of KD, but I think it's super duper important in terms of things like antibody antigen interactions, and things in terms of enzyme interactions, and things in terms of like pharmaceutical drugs. So often pharmaceutical drug makers are going to make all sorts of modifications to molecules in order to try to get a lower KD. Because remember that means that you have to give less of it. And so the patients don't have to end up taking those giant pills 50 times a day if you have something with a higher KD. But you wouldn't want something with too high of an KD. You wouldn't want something with an affinity like avidin and to biotin, because, well, you typically don't want things to stay permanently stuck. But that's hopefully this kind of helps you understand the importance of the KD. So now let's do a little concept check and look at some actual data and interpreting it. This is just a random article I found where they're studying a transcription factor, which is going to be this protein, and they find that it can bind to, so it normally binds to DNA in order to control when the DNA gets made into mRNA. But they found that it can also bind RNA. And so here, what they're doing in this paper is basically they're testing different bindings. They're testing different ligands. So remember, a ligand is a binding partner. And here, if we look at this figure, what they're doing is they're testing double-stranded DNA with a binding site, double-stranded DNA without a binding site, single-stranded DNA with a binding site, this long non-coding RNA, and this other single-stranded RNA. So basically, they have some RNAs, they have some DNAs, and then they go and they measure the binding. They plot the binding. You can see that we are in a log scale here, which is why we get these sigmoidal curves. And now I want you to interpret this graph. So pause the video and answer the following questions. Which of these is the strongest binder? And which ones are the weakest binders? Which would have the highest KD? Approximate those KDs from the graph, and then tell me which would have the highest Ka. Hopefully you did that all, and now let's go over the answers. So which of these is going to be the strongest binder? Remember, the strongest binder is going to be the one that you need less of the other partner in order for half of it to be bound. It's going to be the thing with the lowest KD, and it's going to be this, in this case, it's going to be the black one that's on the left. This paper, like this graph is really confusing in that they have this, like, they're double-stranded DNA with a binding site, and they're single-stranded DNA with binding site. Basically, they're both using these black dots, and even though it shows that they're supposed to be like in this like unfilled form, it looks pretty filled in here, but there's a dotted line. So I'm assuming that this is going to be their double-stranded DNA with the binding site. So the strongest binder is going to be this one. The weakest binders, well, I kind of gave a hint. Basically, they're kind of all overlaid. And so what we're going to have is we had this one, we had this one, and we had this one. These were our weakest binders. And then again, our strongest binder was going to be this one. Okay, so which of these would have the highest KD? So remember, higher KD would be weaker binding. And so these ones are going to have these would be our weakest binders. These would have the higher KD. We can actually go and we can approximate the KD from these graphs. So let's go ahead and do that. Remember that the KD is going to be the concentration at which half of it's bound. So if we go and we take this half Bmax, so remember we can call this our Bmax, and this would be half of our Bmax. Now in this case, it's normalized fraction bound. So they have their Bmax is actually at 1. Sometimes what you'll find is that when you actually go and measure things, you'll find that the Bmax is actually going to be less than 1. And basically this could indicate that some of what you think is like there is actually not functional, or your concentration is a bit off because your purity is not entire or something like that. So your Bmax might not actually plateau at 1, but then you can normalize so that it does. Okay, so now what we're going to say is we're going to go down, we're going to find where the half Bmax intercepts our curve. In this case, what we're going to get is we're going to get something in between 1 and 10 nanomolars for the strongest binding, the double-stranded DNA site. And then in the case of our weaker binders, they're going to be somewhere between 100 and 1,000. Okay, so which would have the highest Ka? So the higher Ka, let's think about this. Remember that Kd is the inverse of the Ka. Ka is going to be the binding affinity. So basically what this is going to mean is that if you have a higher K, that means more of it's going to be bound. So remember Ka we can write as the concentration of AB over the concentration of A times B. If you have more of it married, when you go and take a look, this is going to be a larger number. So if we have a lower Kd, we're going to have a higher Ka. And if we have a higher Ka, we're going to have a lower Kd. So once again, what we're going to see is that the highest Ka is going to be our double-stranded DNA binding site. Let's go back and talk about why Kb is the concentration of A at which half of B is bound. So remember that we could kind of talk about that fraction of B bound as that theta B. So this is our theta B. And basically, if we think about, okay, well, what does that really mean? It's going to be our bound B over our total B. So concentration of AB over the concentration of free B plus the concentration of the AB. So unbound and bound. And now let's think about KD. So KD is equal to the concentration of A times the concentration of B over the concentration of A times B. So now pause the video and use these two equations, put them together in order to solve for the fraction B bound. So solve for theta B in terms of KD. So with a little math, hopefully that you figure this out, where you can basically rearrange things and find that our theta B is going to equal to the concentration of A times over the concentration of the KD plus A. So now what would happen when the concentration of A was equal to your KD? Well, here you would then replace the concentration of free A with KD over KD plus KD, and that would give you 1 KD over 2 KDs, or 1/2. And so that's why the KD is going to equal the concentration of one partner at which half of the other partner is bound. But what if you're not at 1/2? We can use the same equation. We can use the same concentration of A over the concentration of KD plus A in order to find the concentration of B that is bound at any time. So what happens when your concentration of A is 10 times the KD? Pause the video and answer the question. Hopefully you found that about 0.91, so about 90%, over 90% of B is bound to A when the concentration of A is tenfold above the KD. let's go the other direction. What if your concentration of A is only 1/10 of the KD? Pause the video and try this. Now you find that only about 9% of B is bound to A when the concentration of A is tenfold below the KD. Bottom line, the binding affinity matters, and just because things can bind doesn't mean that they will. So when we do binding assays, we can basically figure out a KD for things that have really, really, really crazy high KDs, meaning they're really, really weak interactions. And so we could calculate a number, but that doesn't tell us that that it's physiologically relevant. If that much stuff is not around, you might not have very much of it actually be bound at all. Of course, you also need to consider that you don't just consider the total concentrations of things in cells, because there's things like scaffolding proteins that can kind of help group things together. So local concentrations might be higher. But if something is not present in a very large amount, it better have a really, really high affinity in order to have any chance of it being bound. You might be wondering, okay, well, what about the concentration of B? Remember, we just chose A and B like randomly. So we could be talking about B in terms of A, or A in terms of B. And so basically, if we take this KD equals concentration of A times the concentration of B over the AB, we do this, but instead we do theta A. What we get that theta A is going to equal AB plus the concentration of A plus the concentration of AB. And what this is going to equal is, if we do the same sort of math, we're going to get that our theta A is equal to the concentration of B, our free B, over the concentration of the KD plus the concentration of free B. So we can use either A or B to figure out the proportions. So now let's talk about how we can actually measure binding affinity. So as I mentioned before, we'll often do a serial dilution of one binding partner and have a constant amount of the other binding partner. So then what we do is we just go and we mix them, and we see what proportion is bound at each of these, and then we can go and we can calculate the KD. So remember, if you have none of your A, you're not going to have any bound. And if you kind of have more A than you have than your capacity, then what's going to happen is you're going to saturate. But somewhere in the middle, you're going to pass that halfway mark where half of it is bound, and that is going to be the concentration of A at which that happens is going to be the KD. So there's a slight technicality in that, I said we wanted a low concentration of one of the partners. Well, this is so that we can make a simplification. So remember that when we talk about our KD equation, we're talking about the concentration of free A times the concentration of free B over the concentration of A times B. What we're going to be measuring is the proportion of B that's bound. And we're going to know how much A we put in, but we don't want to have to actually directly measure how much of the A is bound and how much of it is unbound. In order to make our lives easier, what we can do is, if we have a concentration of A that's going to be way, way, way above the concentration of B, then basically any A that gets taken away is going to be a very, very tiny amount. And so what we can do is we can simplify things and say, okay, well, the total amount of A is going to be equal to the free amount of A, because we're taking away just a little tiny bit. So we can just use that total A and don't need to worry about accounting for the B stuff. But as we saw before, if we do have a concentration of our B that is going to be greater, that's closer to our KD, well, then we're actually going to get a lot of our A bound. So we are going to have to take that into consideration. Remember how we saw that if we're tenfold under the KD, so if we're at 1/10 of the KD, we had about 9% of it bound. But that would be about 90% of it is still free. So we're changing about 10%. Ideally, what you'll be is about 100-fold. And so now you're going to have very, very little of your A bound. And so that's why we want to use a low concentration of one of the binding partners. Unfortunately, if you have a really strong binder, this can be really, really hard to have that low of a concentration and still be able to detect it. So often what we do is, if we're wanting to measure, say, like RNA or DNA binding, we'll actually use radio labeling. So I did this a lot in grad school, where basically you can use a radioactive version of phosphorus to to basically label RNA or DNA, and this is going to be super duper sensitive. So you can use that little amount and still detect the signal. Okay, now let's actually go and let's think about where are these numbers actually coming from? So we see we can measure them experimentally, and I'll show you some methods we can use to measure them experimentally. But why? Why are these values what they are? A key hint comes if you look in that table we were looking at to tell us about some of the common examples and range of affinities. So basically, what it tells us is that a reported dissociation constant is valid for only particular conditions under which it was measured. KD values for a protein-ligand interaction can be altered, sometimes by several orders of magnitude, by changes in the solution salt concentration, pH, or other variables. So when we're talking about those interactions, when we're talking about the forward and reverse rate, when we're talking about these equilibrium constants, it's all coming down to thermodynamic principles. It's coming down to the change in the Gibbs free energy. So remember, a negative delta G is favorable, a spontaneous reaction, and the positive delta G would be unfavorable. And the delta G is going to depend on a change in enthalpy and the change in entropy. So remember that the change in enthalpy, this is going to be related to bonds and interactions. And then the entropy, that's going to be our randomness and disorder. When we're talking about this non-covalent binding, what we're going to be dealing with isn't like actual covalent bonds breaking and being formed, but we're going to have a lot of different intermolecular forces, intermolecular attractions we need to take into consideration. So remember that basic principle that opposite charges attract and like charges repel, and this is true of partial charges as well. Even those temporary ones, even those induced dipoles, even those instantaneous dipole-induced dipole or dispersion forces, all of those van der Waals forces, all of those are going to count towards our enthalpy. What's going to happen is that if we have more favorable interactions in the bound state than in the unbound state, that's going to be favorable in terms of the enthalpy. We also, though, if we have more entropy, that's going to be favorable. And so typically, if you were thinking about something binding, you might say, okay, well, the entropy is going to decrease, right? We're taking these two things that could be floating freely around and we're bumping them together. But you have to think about, what are you displacing? And so basically, we have to think about that hydrophobic effect, remember? So the hydrophobic effect is also going to be a key force in driving binding reactions and providing binding energy. So basically, the energy you get by having a more stable or more happy, more energetically favorable in the bound form than the unbound form is often coming from, driven by entropy in terms of the entropy of the water that you're releasing. Binding sites are often hydrophobic, not always, but there are lots of hydrophobic binding sites. And what happens is that if there's not something bound, the water is going to have to be lining them. The water is going to have to be taken out of commission because it's going to try to maximize its water bounds and minimize its interactions with those things that don't offer it anything attractive. So if you had two nonpolar things floating around, or if you had that nonpolar site and you had like a hydrophobic molecule, a hydrophobic drug, say, if water had to surround each of those, you would that would like decrease entropy. That would have a low entropy for the water. But if the molecule were to bind, well, now you would free up some of the water that had been used to kind of to surround those things before, and now you're going to get an increase in entropy driven by the water. So when you have the binding, you have contributions from both the entropy in terms of the water that you're releasing, as well as the enthalpy, because you could be making more favorable bonds, either with the two molecules that are binding. They could make favorable interactions that are also going to contribute to a favorable binding affinity. But as we saw on that table, those affinities are going to depend on the context and depend on the surrounding. So we can kind of simplify things by thinking of all interactions as being like electrostatically based, so charge based. So maybe it's a full charge in the case of our ionic interactions. Remember, we often call those salt bridges. And sometimes it's going to be just like a partial charge thing, either a permanent thing, like a permanent dipole, or a temporary induced dipole, or van der Waals, or London dispersion forces. All of that you can think about as kind of at heart being some sort of charge interaction. If we think about salts, well, a salt is just a neutral combo of ions. So we have a positive ion and a negative ion. And if those are floating around in solution, well, now they're kind of offering distractions. So this means that there's a lot of salt around. Basically, this can increase your KD because there's more things to distract you. But if you have a less salt around, well, now there's fewer things to distract you. There's fewer things to kind of form bonds to instead of the other binding partner. So your other binding partner is your better bet here, or at least you don't see a lot of the ions to bind to. And therefore, what's going to happen is you typically have you have a lower KD. And so often when you go and you do binding experiments, or you're trying to interpret someone else's binding experiments, you want to think about, okay, well, what's the salt concentration? Because you need to make sure that there's not a really low salt concentration, which would make it so that things would seem to be tighter binders than they were. And if you want to see if something's really, really tightly bound, you often do a high salt wash so that you can wash off anything that's not bound very strongly. So bottom line, KD is a measure of the binding strength, but it's actually an inverse of your binding affinity constant. This means that a lower KD is going to mean a stronger interaction, and a higher KD is going to be a weaker interaction. The strength of the interaction is going to depend on both K on, kind of like your probability of binding, and your K off, or your probability of unbinding. Remember that the K on and the K off aren't going to change, except if you change the conditions. And the KD is going to be equal to the K off over the K on. And since those aren't changing, the KD isn't going to change either. So our KD is going to be a constant. And it's this constant, this equilibrium constant, where after equilibrium, instead of defining KD in terms of our rate constants, we can define it in terms of our concentrations. So we can say the concentration of free A times the concentration of free B over the concentration of A times B, or the concentration of the bound AB. So we found that the KD is going to be the concentration of one partner at which half of the other partner will be bound at equilibrium. Remember that the amount that's bound is going to depend on the concentration, because you need to have enough molecules of them around so that you're able to see them in order to bind to them. But you are not going to change the actual KD when you change the concentration. So the KD stays constant. You can then measure the amount that's bound at different concentrations of the other binding partner, and this is going to allow us to determine that KD experimentally. Remember that something can have a lower KD, so a higher affinity, either because it binds more tightly, or like binds more quickly, or because it comes off less frequently. So a higher K on, or a lower K off, or it could also be some combination of the two. Once you're measuring at equilibrium, you don't know anything about that. Some of the methods that I mentioned are actually going to be able to tell you those thermodynamic, those kinetic constants, those kinetic parameters, your K on and your K off. But if you're just measuring things at equilibrium, you're not going to know. Remember that the kind of common range for binding interactions is going to be nanomolar to micromolar when we're talking about biochemical things. In terms of drug design, you don't want something, you often start with something that has a lower affinity, a higher KD, but then you try to work down to something more in that range. So I hope that helped you understand and appreciate KD. And hopefully you are ready to interpret some curves and interpret some data and do that sort of fun stuff.